Sir, we will start now.
Yeah.
A very good evening to everyone. So, on behalf of DDH training team as well as Director CSR NIS, Dr. GN Shastri, we welcome you all for the Vertical 2 session. So today Vertical 2 session will be delivered by Dr. Parthiban Srinivasan. Dr. Parthiban Srinivasan is an adjunct faculty at Indian Institute of Science, Education and Research, Bhopal, and he is mainly teaching machine learning and artificial intelligence. Dr. Parthiban holds dual master degree, one in science and another one in engineering, and he is PhD in computational chemistry from Indian Institute of Science, Bengaluru. After his PhD, he continues his research at NASA Ames Research Center in USA and Weizmann Institute of Science in Israel. Then he worked at AstraZeneca in the area of computer-aided drug design, especially for tuberculosis. Later, he headed informatics business units in Jubilant Bioscience and then in GVK Bioscience before he floated the company Parthis Reverse Informatics. And now his recent venture is Vinyani, a data science company with a focus on AI and ML guided drug design and health. So over to you, sir.
Thank you very much. Thank you very much. Good afternoon. I hope you are able to hear me clearly, right? So yes. At the outset, I would like to thank the organizers for inviting me to this wonderful program, Drug Discovery Hackathon. And today I'm going to talk about big data in drug discovery. That is how the data is created, managed, and how we extract the value out of the data. So that's the concept that I'm going to talk about this on in big data in drug discovery. I believe humans have discovered solutions to most of the pressing problems. But the challenge is the solution comes in the shape of puzzle pieces. A patent document here, a journal article over there, a conference abstract, clinical trials document somewhere, and Chinese internal document somewhere. So all the knowledge is scattered. The abundance of knowledge that we have today is unprecedented. And our brain cannot read all of them. And for instance, every single day, there are about 5,000 journal articles in the area of science, technology, and medicine are published. 50% of those articles are not even read by 5 people. They all end up as digital dust. What if we have a brain, a super brain, which can read all of them, collect the data, connect the dots, and make sense out of it? That is what we are trying to do with the use of artificial intelligence. You may wonder, so what is something new which has come in the form of AI? But actually the concept is not necessarily completely new, but what is new is the convergence of 3 factors. Number 1 is the big data in drug discovery, which was not available for any end users like us, like 10, 15 years ago, or even more than 10 years ago. And second thing is these algorithms are very well matured and developed, and they are all available as open source, which was never there prior to 2016. And then with the availability of the GPU computing, TPU computing, and Colab, things like that. So the convergence of computational power, the big data, and the algorithms. This convergence is enabling all the big pharma companies to advance in AI for drug discovery with the use of big data. So now we know why we are here. So the coronavirus issue, which we have been talking about for weeks, months, and it's almost like more than 6 months now. And given this, every research community, they are moving towards finding a remedy solution towards this. So the AI steps up in battle against this COVID-19 as well. So for those working in this field of AI drug discovery, there are 2 options. when it comes to coronavirus, because we do not have time. You all know that any drug discovery takes about more than 10 years and $2.6 billion and so on, but we do not have time. So the two areas by which the AI community, they are pitching in with the use of big data is, one is repurposing the drugs, and second thing is finding a novel compounds within days, like 20 days or 4 weeks and so on and so forth. So here, if you are working in AI for drug discovery, I would like to mention about two leading companies, which are all pretty recent companies. The number one is Benevolent AI, and second one is in silico medicine. So the Benevolent AI has taken the second option, that is repurposing the existing drugs, whereas in silico medicine, they have went with the designing novel molecules for this COVID-19 issue. For instance, the Benevolent AI, they partner with Eli Lilly, and there is a drug which is already there with Eli Lilly, baricitinib. So that was essentially a rheumatoid arthritis drug. Now that particular compound is repurposed as a promising treatment for COVID-19, and the work is still in progress. So this is one possibility, that with the use of AI, analyze the existing drugs and repurpose it. The second option is designing completely new molecules, making the machines to dream for novel compounds. You know the biological target, especially in COVID-19 now, and you have thousands of compounds available for antiviral compounds as well as protease inhibitors, which are all existing freely available by a click of a button to you. So with the use of these existing big data on a specific biological target, you train the neural network and make the AI platform to dream for new compounds, novel compounds towards the coronavirus. So that's the second approach. So given this, there is a lot of people think that it's not just a hype, it's already a success story. So that's one of the reasons now the pharma industries are investing heavily towards AI. For instance, the pharma market, the AI for drug discovery market in 2018 is about not more than $2 billion. And the projection right now is by the year 2025, within next 5 years, the market is AI for drug discovery is going to exponentially grow towards $35 billion. So this is the current the market research on this. So with that introduction, now let's jump directly into the actual big data and AI kind of stuff. So let's start with a very simple mathematics to understand what the machine learning is and how this is playing a role in designing the new drugs. So we all know that when I say 3 plus 3, and you would say answer is 6. Whether you start counting from the left side or right side, 3 plus 3 is 6. There is no doubt about it. And let me know if anybody disagree with these the elementary mathematics. 3 plus 3 is 6. Now, if I ask you, give you some examples like 1 plus 1 is 11, 2 plus 2 is 22. Now, if I ask you a question, 3 plus 3, and I'm sure many of you would not give me the answer 6, and you would say 33. So what has happened? What has changed now? For years, we have been knowing 3 plus 3 is 6, but now what has happened now? So 3 things happened. I gave you examples, 2 examples, and you built a logic in your mind. You built a logic in your mind. Now, when I give a third test case, you applied that logic onto the new test and you came up with an answer. So this is essentially a rule-based approach. So when I look at it, you can build a logic. That's the rule-based approach. So you can build it. But now the first set is OK. Now, if I give you another set of problem, 1 plus 1 is 10, 2 plus 2 is 20. Now 3 plus 3 is not 33. You will say that 30. Likewise, 1 plus 1 is 3, 2 plus 2 is 5, 3 plus 3, what? It's again, you will say 7, different answer. So we cannot build every new set a new rule. We cannot continuously keep on building a rule. What we need to do is we want the machine to look at things and build a rule by itself. So the difference between a traditional programming and machine learning is that there is an input and there is an output, and in between there is a recipe. That recipe, who is building it? The model, who is building it? The function, who is building it? That's what makes the difference. In traditional programming, we, the humans, we give the rules, set of rules. Whereas in machine learning, the machine builds the set of rules. So that's the difference between the traditional programming and machine learning. And to make it much more clear, here in the traditional programming, we kind of send the rules and data. So the rules and data goes in and answers come out. And we express those rules in programming language. Maybe that is Java, C, C++, or Python or whatever. And that is what is traditional programming like since day one. The machine learning revolution flips the axis as shown in this diagram, the green color. Instead of rules and data coming in and answers coming out, we now feed in answers and data and rules come out. So the whole idea behind machine learning and the whole revolution of machine learning is this diagram. Now, what does this mean? How does this apply to real system? So let's think about if you are writing a code for activity recognition. So what you see here. So you are wearing a device, wanted to determine if the person is walking. If it could have a speedometer or accelerometer. You could say, well, if the speed is less than 4, then he's probably walking. OK, if you have some naive algorithm, but it works. So what about if he's running? So we can check again with the speed. And if the speed is more than 4, then probably he is running. It is still a naive algorithm, but it works. And then we say, what if we want to add biking to that, cycling? Now we say if the speed is certain amount, probably walking, and if it is less than certain amount, is running, and if it is more than certain amount, we can say that the person is biking. So that's clear. This is again a naive algorithm, simple rule. It's pretty clear. It's not too difficult. But what about this? So what algorithm can identify this particular activity? So this is where the limitation of the traditional code comes in. So how do we solve this and how do we fix this? Let's go back to this diagram. So machine learning is all about feeding answers, feeding data, having the computer to figure out the rules and which matches those answers to the data. Then we can say the activity recognition may be looking like this. So we gather data from the materials attached to the person, and we say when she is walking, this is how she looks like. When she is running, this is how she looks like. And when she is biking, this is how it looks. And when she is golfing, this is how she looks like. This opens up a whole new scenario. Think back to the name algorithm earlier. So different people play golf at different skill level, gather enough data. Now you can make out the patterns. This is what is walking, this is what is running, this is what is biking. So here the machine learning is really the key to that. So essentially, you can make out the patterns from the different activity. So the key to getting all this is the data and figuring out what matches all of these data from all these people to these particular outputs. So let's take a very simple example. Here is the X, which has a certain value, and corresponding Y has certain value. Can you think of mapping the relationship between X and Y? So one way to look at is in X, all the values increase by number 1 and Y increases by number 2. And when I look at when X is 0, there is -1. So probably you can arrive at, by looking at these numbers, you would arrive at a sort function, which is Y equal to 2X minus 1. So how did you get this? Now, likewise, if I give you another example, like the same fashion, -1 and blah, 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 blah. So here for this pattern, by analyzing that, we can come up with the solution as Y equal to 3X plus 1. So how do we put this into programming? So this is what matters the numbers to each other. So this is what we are trying to work on this. So this is the code which can do the using TensorFlow and Keras. And if you are already working in AI, you probably might have heard about what is called TensorFlow and Keras, PyTorch, and the neural networks essentially works. Um, uh, we give an analogy, something like how the neurons in our brain works. So essentially the electrical signal and the chemical signal which passes the information from one neuron to another neuron in our brain. There are trillions of neurons in our brain. So the input signal goes into the neuron and there is an activation function which gives the output. So for our today's discussion, I am taking a very, very simple neural network. So what you see, the green color circle, which is the neuron, and it receives the input and gives you the output, which is if you would have seen some of the neural networks with the complicated structures with the multiple hidden layer, one input, several hidden layers, and then one output. Here for our discussion, I'm considering only one hidden layer, so which is the green color circle. And also to make the discussion very extremely simple, I'm considering only one neuron. So every layer can have hundreds of neurons. Every neural network structures can multiple hidden layers. So in this example, I'm taking one hidden layer with one neuron. So that is what in the first line, when you read, we are defining the model. Model equal to keras.sequential, keras.layers.dense, units equal to 1. So which means units, I'm taking only one unit, and this one first layer which defines the first hidden layer. So one hidden layer with one neuron. So that is how I'm defining the whole neural network here. So the key part is the second line, model.compile. So now I'm compiling. So you would have seen that artificial intelligence is more of mathematics, linear algebra, calculus, probability, and statistics. So where does it all comes into? So that's what the second line. So it encompasses all those complicated mathematics. So it has two key functions. Number one is optimizer. Number two is loss. So the optimizer is nothing but the learning. So when you say machine learning, deep learning, so where does that learning comes from? So that is what is the optimizer. SGD here stands for stochastic gradient descent. There are several options available for choosing the optimizer. And here for our today's discussions, I've just taken the SGD, stochastic gradient descent. And loss is essentially when I am making a guess. The optimizer is nothing but I'm making a guess. So if I am guessing, let's say that the distance between Chennai and Delhi, I'm kind of trying to think, my first guess is, let's say that 20,000 kilometers. But then the actual value, all of us know. So from the original value, when I subtract it, so that's the error. So with that error, all I know is some error which is there. So then now I try to make the distance as 500 kilometers. Oh, now I'm getting an error in an opposite direction. So the loss function here is mean squared error, which tells me, hey, you have error with this level of quantity. So that's what it tells me. With that feedback, the optimizer SGD makes a new guess. So every guess should give error, lesser error. As I keep on doing this again and again, the error value should go down. So at the bottom, you see epochs equal to 500, which means that I'm telling this program to make 500 iterations. So with the 500 iterations, that I'm trying to find a solution for this. That is the data that I'm giving in the middle, xs and ys. So here NP is a NumPy array. So x values and corresponding y values I have given now. And tell me what is the function, what is the relationship between this x and y? How the x and y are mapped. So this is what my problem, and this is what I want to do it. So that is what I'm asking in the next line, model.fit x, y's. So this is my input and this is my value for that. Do it 500 times. Once you complete the model, then I have a new value. Predict the y value for a x, which I am assigning it as value 10. So 10 is x value. Now you tell me what is the value of y. So one of the reasons why I'm elaborately discussing this particular part is essentially it has, if you closely look at it, it has 5 components. The first line is the first component, which is defining the model. Second line is compilation of the model. Third component is the data that we are feeding in. Fourth component is fitting the model, defining the iterations. The fifth component is prediction for the test case. So this is the hello world of machine learning. So this is the most simplest neural networks that anyone can think of. Another reason why I am emphasizing this particular slide is whatever be the complicated neural network that you can think of today, whether it is a computer vision problem, object identification, object recognition, or natural language processing, you name any of them, all of them can be divided like this 5 portions. So this is the core concept which is expanded into any possible artificial intelligence problems. And the same stuff, the components of what you see in this slide is exactly precisely needed for predicting the biological activity of the data. For instance, when I have a set of molecules for which I know whether they are toxic or non-toxic, it's a binary classification. Or if I know there are 24 set of compounds for which I know I have a biologically active, moderately biologically active, biologically inactive. So this set of input, I do have it. Molecules and their bioactivity data, I feed them and then build a model out of it. And then I'm trying to predict biological activity of unknown compounds. So the whole concept comes into this. So probably for each one of those problems, based on the problem, the number of lines, the complications might be more, but the concept is what you see here. This principle remains the same in any problem. So when I run this code, so I'm asking, if you look at the top layer, so the epoch first 1, 2, 3, 4, 5, 6, that's at the beginning. At the right side, you can see the loss is 44, 34, 27, 21. It starts decreasing. And at the bottom, you see that epoch is 500. So at the 500th cycle, it is like 3 into 10 to the power of minus 7. So which is like a 0.0000 loss. So if you look at this, so here it is clearly the 3x plus 1 is the equation that it's trying to map. And it is building an approximate model. And if you look at it, it is not exactly giving 31. It is 31.000175. Even if you run it twice, thrice, you will get something like this. The one of the reason is they're all approximations. We very well know as it's a linear function. It is clear cut 31, but still, there is a lot of probability which is involved in this in a decision making process. And also, the points might be linear at the earlier points, minus 1, 0, 1, 2, 3, 4. Here it may look linear, but when you are extrapolating it to 10, it need not have to be, things are as simple as a straight linear function. And the neural networks, the neurons are very complicated with the nonlinear functions, the activations functions which are involved in that. And it needs to take probability and approximations into account. So that's one of the reasons that you are not getting 31 exactly. So you get either 30.999 or 31.0075. So likewise, the answers would be like this. So that's kind of an introduction about how the big data, which can be used into the decision-making process, machine learning, artificial intelligence. Now, can you recognize the picture here? And he is Christopher Lipinski. If you are working in medicinal chemistry or computer-aided drug design, you should know about this person, Christopher Lipinski. He was working as a senior scientist in Pfizer company. And in the year 1997, so he went to the pharmacy and he looked at all these drugs. What is common in these drugs are all of them are FDA approved drugs. So all of them have cleared all the FDA struggle. And you all know that the drug discovery has several milestones. And these drugs have crossed all those milestones. And all these are all tablets, which means you are consuming. That is, all these drugs are, whether it's a fever or a headache or a diarrhea or any of the issues, these tablets, what you do is just you are putting it in your mouth and then consume a couple of glasses of water. So oral bioavailability is one big issue all these drugs have cleared, which essentially means there must be some common factor. There has to be some pattern which needs to come out of these drugs. So he started analyzing the data in terms of molecular weight and the water octanol coefficient and all that. So when he looked at these data, he found a pattern, like molecular weight for all these drugs, most of them are less than 500. Log P is less than 5. And number of hydrogen bond donors all these drugs are having are less than 5. So which is essentially the functional groups of NH and OH. And number of hydrogen bond acceptors are less than 10, the sum of nitrogen and oxygen in the molecule, which essentially accepts the hydrogen bond electrons. So this is what he came up. So since all of them are multiples of 5, they call it as the Lipinski rule of 5, or some people say that the given new molecule should at least fulfill any three of these conditions. So that's the 5th condition, and some people say. So whatever, but again, you need to keep in mind, many scientists criticize about this rule of 5, but at the same time, everybody uses this rule of 5, and they do respect this rule of 5. For instance, when you are in lead identification stage, you have a hit molecule, you have a lead identification stage, and from lead identification, the molecule has to move to lead optimization. Essentially means you have a biologically active compound, but at the same time, during the lead optimization, it should be free of toxicity, permeability, and various other factors. It has to be manufacturable, scalable, and various other factors the molecule needs to satisfy. So in order to satisfy those conditions, you start adding a functional groups. For instance, solubility factor, you want to improve it. Okay, instead of a methyl group, you may start increasing methyl, ethyl, propyl. Instead of that, you still keep increasing, but maybe after a certain point in time, after 5 carbon atoms in the side chain, you may think that it doesn't improve, so you stop over there. So likewise, you start, kind of a play around like a juggling of various balls in this. So when you start adding functional groups, typically a thumb rule is the molecular weight increases by 75, which essentially means when you are starting a lead identification, you should not start with the molecule with the molecular weight 500, because when it moves to lead optimization, anyway, it's going to increase close to 75 molecular weight. So which then it starts violating the Lipinski rule of 5. So we got a lot of a clear understanding through this, and these all came out of just by simply analyzing the data. So that's the power of analyzing the data. So now let's look at the problems with the data, what we do encounter. The problem, see, the machine learning is fine. Machine learning brings the magic. Okay, let it be. But the magic can happen only if the data is in a consumable manner. So if the data is not consumable by the neural networks, it cannot produce the results. So most of the time, as a computer-aided drug design scientist or data scientist or a software programmer, whatever that you may call, the problem that you will be assigned to you most of the time would be cleaning the data. And as a modeler, you would also find these are the problems: insufficient data, too much data. Too much data essentially means it's most of them are garbage. Most useful information would be sitting in very tiny little fraction. And so no, most of the time you would be end up in extracting the jewel out of junk. So that's what I mean by too much of data, non-representative data. So that doesn't make any sense for us. So just because you are having a large amount of records doesn't mean that each one of them has to represent the problem that you are working and solving. And that very many times missing data. So here I'm not talking about science. I'm talking only from the software programming point of view. So when you are having a missing data, how are you going to extrapolate it, or how are you going to throw it? So when you are given a Excel sheet with the 10,000 records, first you need to analyze it, and then wherever there is a missing data, you need to have a policy on handling such records, whether you throw it away or you can kind of extrapolate with the use of some data available. So and also most of the time it will have a duplicate data. And you need to remove those duplicates and outliers. So outliers, sometimes it will be misleading. So these are the problems that we normally handle when you are handling the big data. So as I said, the big data, when I say big data, there are two ways to look at it. The credit card database in a given city let's say there are 1 million people, they are using credit card. So how the credit card database would look like then? It will have a millions of records. For each record corresponds to one person, but for every person, the record will have a not more than 10 columns. Column 1 is the name of the person, the credit card expiry date, and probably some of the other hidden information, and the card in which the on which the card has been released, and the address. So likewise, hardly not more than 10 columns, but it is a very tall but a narrow database. Whereas if you look at the drug discovery database, here most of the time we may not be handling more than 10,000 records. But if you look at the number of columns, it might run into hundreds. Number of features for every single compound. If you use any of the descriptor calculations, some of them are Dragon, those kind of descriptor calculation, the platforms, they give close to 1,000 columns. So that again, handling that is extremely difficult. Essentially, you need to understand what is the principal component analysis. What I mean by different column is each one is a different dimension. So human brain cannot perceive anything beyond 3-dimension. We are living in the 3-dimensional space. But if you keep on adding another column, which you are handling the data in a multi-dimensional space. So when you want to present the results in your laptop, you have to reduce those 100 columns into 3 axes. So it's a kind of a dimensionality reduction that you need to do it. So these are all the problems that we face. How are we going to work on this? So this is what the main crux of the talk. So essentially, so this is what everything comes under the domain of data science. So data science is essentially the discipline of making the data useful. So when the data is in the useful format, you can combine the data from multiple other sources, integrate it, and extract the knowledge out of the integrated database. So here I am giving you a simple example. Typically, we conduct experiments and then we publish the results in journal article. So which essentially means that if you're working in specific, let's say COVID area, so you probably the protease might be of your interest. So where do you start your research? Where do you start your model? You don't need to start anything from scratch. There are hundreds and thousands of journal articles have been published, hundreds and thousands of patents have been published exclusively on protease. But you do not have time to go back and then read every material. So some of them are already available online, open source, free of cost. On a click of a button, you can pull out those information. So how they are prepared, essentially from the journal article, someone reads it and then puts it in an Excel sheet and then convert that into a database. So here I have just showed the example of a biocuration process. So by click of a button, the user would be as a modeler, as an AI person, or as a computer-aided drug design person. By a click of a button, you would be interested in looking at all the molecules that have been reported to be interacting with protease. So this is what the purpose of the big data in drug discovery. So I don't want to spend my time on reading all the thousands of papers. But again, the solutions to some of our problems are already sitting somewhere in the billions of pages of documents, literature. For instance, you can Google about somebody called Swanson. Swanson was a librarian. He was at the end of the day, every day he used to keep the books at the rack. So he was just casually flipping the book, and then he was started reading somewhere that the Raynaud syndrome. Raynaud syndrome is essentially your fingers, your palm, they become pale, so colorless kind of a stuff. So that's mainly because of the change in viscosity of the blood. And he was just glancing through it. And then after a couple of weeks, he was also reading another book where the fish oil could alter the viscosity of the blood. So he just that's one dot, this is another dot. He just connected these two dots. He has not done any experiment. He's not a life sciences person. So fish oil could alter the viscosity of the blood. On one side he was reading, and the other day he was reading Raynaud syndrome is mainly due to the change in viscosity of the blood. So when you connect these two, fish oil could be a remedy for Raynaud syndrome. And today that has been proved and the FDA has approved. So the idea comes by just connecting the dots. So all you need is an effective algorithms. So that's what we call it as a machine learning. The purpose of that is connect the dots, make sense out of it, and enable the drug discovery. For that, we need to have the big data such as like this kinase inhibitors. For this, you need to understand the latest programming ecosystem. For instance, the Python. So Python programming language of late, it is becoming a de facto standard for people working in the machine learning area. So now let's little bit touch upon the science part of it. So the two pillars in drug discovery, the biological target and chemical compound, and they're all connected by bioassays. So whether a given molecule is effective towards a target of your interest that you study through bioassays, and which are all already reported in the literature. And the target classes, normally the major classes are like GPCR, kinase, protease, nuclease receptors, and ion channel. And if you look at any of the patent website, for instance, US Patent Office, uspto.gov, just go and search for GPCR. You will find thousands of patents on GPCR. And you will find thousands of patents on kinase, thousands of patents on protease. So that is where you need to start your big data research. So how do you collect those information? How do you collect those documents? How do you integrate that with your drug discovery process? So that is what would enable us to really exploit the power of artificial intelligence. And the chemistry space, we say that about 10 power 60. So we are trying to okay, so let me put it this way. What is the big difference which is happening in the last couple of years in these computer-aided drug design? Is it something, a new buzz which has been going on? So this, sometimes you may wonder, say in QSAR, I was choosing 24 compounds with the biological activity, and I was dividing a training set and test set. So what's the big deal which is in AI going on? This is what I have been doing it. Are you giving a new name for what I have been doing already? So some people might ask this question. The big difference is generative models. The big difference is generative models. Never ever we were able to dream for new compounds. All the time, all the computer-aided drug design group was doing is predicting the molecular biological activity for a given set of compounds. But we were never, maximum what we could do is generating the marquee structures, nothing more than that. But we were not able to dream for a new compound by training the neural networks with the existing knowledge that we never used to do, were able to do it. That is what is feasible right now, and the success stories are emerging out of that. We will see much more detail on that. So typically you have a biological target and molecules. There are two variables. So when you have two variables, then there are 4 situations. So what are the techniques that you need to choose? So the first part is, you know what is the target and you have a molecule. So all you need to do is whether the given molecule will bind it or not. So essentially, you know both the molecule and the target structure, X-ray crystallographic structure. So this is where you normally do the docking. And this is the biggest piece. This is the change that de novo drug design has been going through a phenomenal change, that is generative models. So I have a target. Can the AI platform dream for a new molecule that can fit into this active site? So that is what the new thing which is going on right now. And we are working on that. So here the third component is, I don't know the target, but all I know is that this molecule is killing the microorganisms. So, or it is it's making a bioactive, showing some activity. So this is where the pharmacophore normally we use it, even if we know the target. And if we don't have a target, especially in the areas like tuberculosis, where we do not have a target, but all we have the information is MIC, minimum inhibitory concentration. So that's where we use a pharmacophore. And then at the beginning of a project, all I do is disease selection. So with the disease selection, I don't have a target information. I don't have a molecular information. So this is where the chemical diversity that we use. So having said all this, I would say that the people say that the evolution of drug discovery is that from in vivo, in vitro, in silico, that is doing experiments with animals, doing experiments in a glass plate, and doing experiments in the computers. So this is how we call. And now I coined a new term in machino, that is using artificial intelligence to dream for new molecule and find the biological activity and also find the toxicity, all that information. So that I call that as a in machino, that is learning from the existing big data and enabling you to come up with a better decision. So that's what I call it as a in machino approach. And look at the picture here. So why the pharma industries are rushing towards AI today. So here on the right, you see the Moore's law. So as the time goes, every 18 months, the computational power is doubling. And everywhere, even in the genomics, if you are working, you probably might know that when we first sequenced the genome, it was close to $1 billion. And now it is hardly a few $100s. So the prices are all coming down. But what is happening in the drug discovery is, like in the year 2000, it was about $1 billion, and then it grew to 1.8. Now today it is standing at $2.6 billion. That is, if I give you about $50 billion, how many drugs that you would be able to discover? So that's a kind of a question that people are asking. So the productivity is going down. So this is what the reverse of Moore's law. This is called Irum's law, because it's all going in the reverse. So one of the biggest reasons for that is it's not science. It's not about, the problem is not just with the science, it's with the decision-making process. Because every stage in the drug discovery is nothing but decision-making. I have an option of 8 molecules, 8 core scaffold. Which one should I proceed? All the scientists, they join and meet in the conference room, and then they start discussing about pros and cons of each and every the scaffold. And then, okay, so we have a budget of such and such money, and we have a time period, which essentially means that we cannot float a project on all the 8 scaffold. We have to choose 2 scaffold. Let's choose 2 scaffold. So one way to choose is that most of the time, the scientists, they don't publish the negative results, but you can infer the negative results by looking at the trend analysis. In the last 10 years, for this scaffold, for this biological target, is it increasing or decreasing? So that also tells the story, but there is something is wrong with this particular scaffold for this particular biological target. So probably a choice, the selection of that particular scaffold may not be a good idea. So likewise, every stage there are decision-making process. So what is machine learning? It is essentially nothing but enabling you to a lot of small decisions. It's not a magic box, but at the same time, it enables you to make a better decisions when you are working in an information-rich environment. So that's one of the reasons chemists pin hopes on deep learning for drug discovery. And this is from an article from Chemical Engineering News. Pharma scientists are exploring what the algorithm behind voice and face recognition can do for chemistry. So on the one hand, the AI is exponentially growing and it is giving a lot of success stories. On the pharma sector, very depressing trend is going on. So this is what you say, big breakthroughs happen when what is suddenly possible meets what is desperately needed. So now I wanted to jump into, for the next few minutes, I wanted to show you how practically how the coding program would look like for you when you really want to work on this. So this is just to show you how the pandas, which has been growing from like hardly 5 years ago, this was one among various languages. And after 2015, it is kind of exponentially growing and it is even surpassing Java, JavaScript, and so on. So Python is becoming a de facto standard language for AI today. And because it has a lot of libraries, which helps you to maneuver around with the process, the big data, and make it enable you to work on various AI algorithms. Now let me stop sharing this. Let me take you to the browser. So I just want to check with you. Are you able to see the browser now? BioIT World. Can you see that?
Yes, sir, we can see it.
Yeah.
Yes, sir, we can see it.
Okay, fine, excellent. So I wanted to give you so where do I start? So some of you might be beginners in this field, some of you might be veterans, but still, if you are not aware of this particular conference website, you should have a look at this. So this is, I've been attending this conference. Usually it happens in the month of April in Boston, but this year, due to the COVID situation, it has been postponed to October, but still it's a virtual conference. So when you started in the way back in 2002, 2003, most of the talks, the tracks are on computer-aided drug design, de novo drug design, pharmacophore, and then the sequencing just started at that time. But genomics were the big thrust areas. 10 years later, that's around 2010, most of them were on bioinformatics and genome sequencing, analyzing the genome sequencing data, microarray, and so on. But now in the year 2020, when you look at it, look at the kind of tracks that you are seeing: data storage, data metadata management, data science and analytics, and also in the middle you see AI for drug discovery, emerging AI technologies, data visualizations. So you might want to click on any of these tracks and then have a look at the various titles and the concepts. So this is where you need to keep track of the areas of AI in drug discovery. Otherwise, you might sit in a corner and then you might think that something, a big thing that you are working on, but somebody else might have already worked on that and then kept it in an open source. So you should not, stand behind kind of even 3 months here is you are outdated. If you are not watching this field for 3 months, you are outdated. So then the second thing that I want you to have a look at is this Campbell database. I'm sure that most of you would have seen this Campbell. So here, if you look at it, this is the COVID-19 database. So this shows the how many biological assay activity data points and then how many number of compounds. So see all visualization. So this tells you about the Campbell data, and it gives you information about how many compounds. So this is what the big difference that I'm talking about. This data was not available few years ago, or 10 years ago, or 5 years ago. And now you have a 2 million data points at a click of a button, which is available to you. So when I was working in AstraZeneca, I didn't have any luxury of this. So when you are building a neural network model, so it is data hungry. Neural networks are data hungry. It gives very good results only if you are able to feed large amount of data. And here you can see that 2 million drug-like compounds are available along with the assay data, biological activity data, and so on. So now let's go back here. Let's look at the browse all Campbell or this one, the explore. Let's look at this particular data. And this is protease database, which is available for you. And you can just simply by clicking here, you can see at the right corner CSV. So you can download these files in the CSV format. So CSV format, I'm sure all of you are aware of it. It's nothing but a plain text file, and which is each column is separated by colon or semicolon and so on. So now I'll show you. So this is here. I've downloaded the Campbell molecular database, and then now I brought it into the Jupyter notebook. So here, what I need is Scikit Learn. I'll show you what Scikit Learn is in a minute. So pandas is extremely essential. Okay, let me take a step back. So essentially, you need to install Anaconda in your system. Anaconda, which comes up with the 275 packages. Like 3 years ago, I had to install each and every one of them manually. But today, Anaconda is so powerful, by a click of a button, you can install all that is needed. All the 275 packages will be installed at one stretch. And this is what you are seeing right now is Jupyter Notebook. I'm sure most of you would be knowing what is Jupyter Notebook if you are working in this programming area. And So here Scikit Learn, import Scikit Learn, which essentially means that I'm importing the machine learning libraries. And when you are handling data, you need to be familiar with what is called pandas library, import pandas as PD. So this reads the data, and then at the top, or here I'm looking at the last 5 records. So the first column is Campbell ID, name, synonym, type, maximum phase, molecular weight, targets, bioactivities, and so on. So if this goes, I think there are about 31 columns here, and the smile strings, the molecular structure in the smiles format is in the last column. So you should be able to first analyze. So when you are looking at when you are given a big data, analyzing essentially before jumping into machine learning or deep learning or AI, first thing is you need to analyze the data that you have, what you have. So how do you do the analysis? There are 2 ways to do it. Number 1 is through the statistics. Number 2 is visualization. So for statistics, you need to know how to handle the data set with the use of NumPy and pandas. The NumPy and pandas are libraries which can be accessed through Python programming. So you need to know Python programming and NumPy and pandas to analyze the statistics of the big data that you have. And for visualization, you need to have a matplotlib, which again can be accessed through the Python code. And here you are just browsing all these various columns and so on. And now from the smile strings that you are looking at it, you are trying to analyze each and every one of them, and looking at the head, that is the top 5 molecules here, molecular information, and then you can pull out, like this is the 99th record, which is essentially 100th records, because here the index starts from 0. 99 means it's a 100th one. So this tells you the shape, which is if you look at the print df.shape, meaning how many columns, how many rows, 6,900 rows and 31 columns. So this tells you the number of rows and columns. So now you have the data. So now also you need to install what is called RDKit. If you want to visualize the molecules in your browser, the Jupyter notebook, so you install the RDKit, which can interpret your smile strings into the molecular structure, as well as the mol file. If you have the mol file, you can import into the Jupyter notebook and you can visualize the molecules. One of the reasons that you need to understand, these are all the ABCs. This is the basic steps. What I have showed is the very, very basic, the first step. So once you achieve this, then with the use of RDKit, which is a very powerful platform, so which will help you to calculate the molecular descriptors. With the use of molecular descriptors, you can do the linear regression, logistic regression, all that with the use of Scikit Learn. So the Scikit Learn is the scikitlearn.org. If you go to this page, it gives you all the details. So whether you are handling the data analysis, is essentially there are 2 things. One is prediction, the other one is inference. So the prediction is whether the biological activity data, so I have 10 molecules with a given biological activity data. So what is the bioactivity data of a new compound? Or whether my new compound is toxic or non-toxic. So these are the kind of information that you are trying to predict for the unknown molecules. So the second area is the inference. So inference is essentially, is this descriptor playing a major role? Whether the given functional group playing a major role in deciding my given molecule biologically active or not. So these are all kind of an inference. To do the entire analysis, you can do the classification, regression, and clustering. And when you are free time, you can just go into this and then you can analyze this. I think I am kind of coming closer to my time for a question part. So with that, let me close this sharing of this part. Let me go back to. Are you sure you want to share? What is the slides part? Okay, I think I'm done here. Maybe we can take few questions here.
Thank you so much, sir. It was really a very wonderful session. And now I request Lizo to please take the question that has come up in YouTube.
Thank you very much, sir, for your most informative sessions. So we have received enough questions on YouTube as well as on MS Teams. So with your permission, we would like to proceed with that.
Oh, you have received some questions. Okay, yeah, go ahead.
Yeah. Yes, sir. So one of the first question I would like to ask you is enumerating the new molecules from the existing chemical data set is one of the challenges that we have in designing some new chemical entities. And for this, the two main components which is required is first of all, we should have some chemical data set, as well as second is the algorithm, the new type of either machine learning algorithm or deep learning algorithms. So what is your opinion or your comments on how the in machino kind of strategy can be applied here for designing new chemical entities?
Okay, that's an excellent question. I'm just trying to see if I can. Do you see my slides? Am I able to share the slides again?
You have to reshare it. Yes.
Share. I'm trying to share the slides. Are you able to see the slides? Can you see the
not
slides?
Yet, sir.
Not yet?
Not yet, not yet.
Oh, God. I think it says stop sharing. Now I'm trying to share. Are you sure you want to share? Yes, share. I'm just trying to share this. No, you are not able to see it. Let me close the browser then, because that's a very interesting question. That's a good question as well. So I just thought I have some slides. I thought I can share the.
Yes, sir. Now we can see. Yes.
You can see the person with the hammer?
Yes.
Oh, okay, fine. Excellent. Let me see if I have that slide. Sorry for. Yeah. So here is the. So this is essentially to represent the kind of making the neural network to dream for molecule. So the first element is how you are going to represent the molecule. Because just by having a molecule doesn't mean that the neural network is like a 3-year-old baby. The 3-year-old baby cannot understand the chemistry, mathematics, and physics, right? So we have to first train the neural network, make it literate on medicinal chemistry. How do you do that? So you have a millions of compounds database like Zinc database, PubChem database, and SureChem database. They are all available online for free. Click of a button. First, you need to represent those molecules by which the neural network can consume that information. So once it is very well trained on chemistry principles, you don't need to teach the neural network carbon has valency 4. But if you send millions of compounds with the carbon compounds, it will automatically infer carbon valency is 4, nitrogen valency is 3, hydrogen valency is 1, oxygen valency is 2. By looking at all the molecules. So for that, the first biggest task is representation learning. What is known as representation learning? Representation learning is, so what you see in this slide right now is the various ways by which you can represent the molecule. So of late, the graph theory is also playing a major role, but still the smile strings are okay to begin with. So you have to ensure that the smile strings are converted into a graphical form and then that is getting consumed by the neural network. So now my 3-year-old baby is very well trained on understanding the chemical principles, the medicinal chemistry principles. Once I have that, it is like generative models is essentially like police and thief. So the thief always try to kind of produce a fake nodes and then he tries to escape. Police always tries to be smart and then catch hold of the thief. So when he comes up with the fake nodes, likewise, these molecules, the dreamed molecules are fake molecules. And you need to create 2 neural networks. It's not 1 neural network. One neural networks generate the molecule. Another neural network is trying to predict whether the generated molecule, whether the molecule is fake or the real one. So the another, which is called generator and descriptor. So let me show you what it is. Here, this is what, I mean, I don't think I'll be able to explain this in a very short span of time, but quickly I can tell you how we do that. On the out of the 4, you can look at the left top corner. So it's like if I have a Word document, when a big huge Word document, when I want to send it to you, first I do that zipping, zip it and then send it to you. And the zip file, we can't make out anything from that file. So what do you do when you receive the zip file? First you unzip it and open the file, start reading it. That's exactly the same principle what you see in the top left corner. I have a molecule, I'm sending it to the encoder, and it is start building the latent space. The latent space is what I call that as a dreaming of new molecule. So within that latent space, the yellow color dot that you are seeing there, that particular dot represents the molecule that you already fed into the encoder. And through the decoder, I'm trying to reproduce the same molecule. So if I can reproduce the same molecule just by looking at that particular dot, what you see on the right side is the reproduced molecule. And the square in the middle, latent space, which is completely empty. Empty doesn't mean that there is no molecule in that. There are molecules sitting there. Only thing is we don't know. So if my encoder decoder, 2 neural networks can reproduce the molecule that I originally fed in on the left side, that if you are able to reproduce, which means that my decoder can decode the empty space. What molecules possibly could exist in that empty space, my decoder can able to pull out that molecule. So this is what is the dreamed molecule. Now I have another neural network, discriminator. The discriminator I train the discriminator with the use of, let's say, the Campbell molecule, Campbell molecular set that we saw right now, just now. So whatever the molecules which are in Campbell, they are all not imaginary. They are all real molecules. They were all synthesized by someone. They were all tested by someone, and they were all published in the journal literature or patent literature. So they're all real molecules. Now my discriminator is very well trained by those Campbell set of molecules. Now my neural network is able to identify when you send me a molecule, whether it is a fake molecule or a real molecule. If my discriminate, so I try to improve the discriminate. Our task is, as an AI scientist, our task is to ensure that the discriminator is smart enough to identify the molecules. At the same time, the generator is smart enough to fake the discriminator. So at some point in time, it's called Nash equilibrium. So both of them try to learn from each other. Discriminator tries to learn from generator, generator learns from discriminator. After a certain point in time, it reaches the equilibrium. That's where we stop the whole coding. So till we reach that position, we try to run the whole code. So that is how we are trying to build a new molecule for any biological target of interest. So this is what right now every pharma industry is rushing towards, making it possible for the disease of their interest. I think that answers detailed answers to your question.
Yes, sir. That was a detailed explanation of the query, sir. Thank you very much. Sir, I'd like to move on to the next question.
Sure.
And it is asked by Ms. Hema Malini, and her question is, can we predict the ligand receptor interactions using AI and ML for any kind of molecule?
Exactly. Wonderful question. So here, this is what the answer for your question is in this slide. So essentially, on the left side, what you see is the chemical compounds. As I said earlier, you need to convert the molecules in a form which the neural networks can consume. At the bottom is the toxicity data, or it could be a so this is a classification problem. The same thing that you can convert this into a regression problem. So the regression, through the regression problem, the regression or classification that you can decide in the last layer. After the hidden layer, the last layer here, how you define whether you are trying to predict the biological activity data or you are classifying a given compound as toxic or non-toxic. That is a binary classification. So that you decide after the hidden layer, the last layer, that's how you model, that's how you design it. But all you need is the inputs. So if you are trying to do a binary classification in terms of toxic or non-toxic, all you need to give the input as the molecular structure and whether the molecule is toxic or non-toxic, the label information. That is what you need to give it. If you are working on a bioactivity information, all your interest is to predict the bioactivity of an unknown new compound. Then you need to give a bioactivity of certain range, like biologically very active, like let's say nanomolar active and micromolar active, millimolar active. So all that entire range that you need to give it as much as possible. And then the molecular structure. So molecular structure and the bioactivity data, when you give it, and you define this problem as not classification, but as regression. So once you define this problem in the last layer as a regression, then you will get the output of for your unknown molecule, what could possibly the bioactivity for the predicted bioactivity for the unknown molecule. So that's how you need to design the neural network. Hope this clears you. Yeah.
Thank you, sir. I'll move on to the next. It's not a question, the next one, but many people are asking about the suggestions or some kind of ideas of if they want to start like a beginner for AI and its applications in drug discovery. So how can they go about? So can you please share your suggestions and your viewpoints here?
Okay. That's a good point. So I am assuming the question was asked by the life science people, not by a software engineer. So I mean, what I'm saying is, so I'm assuming that you already have a domain knowledge in life sciences. You understand what is a biological target. You understand what is a lead molecule. So that is a basic knowledge that you have. Now you want to get on to AI for drug discovery. So that's what I'm assuming, because there are two ways here. One is even the software engineers of late, because today the healthcare drug discovery, they are very much promising. Very many software engineers also wants to jump into it. They want to learn the biology, but at the same time, the biologist who are kind of whether bioinformatician or even the wet lab scientists, they have a sufficient knowledge on the domain. So now they want to get into the AI. So now I am assuming the person who is asking the question. Pardon? So I'm assuming that the person who asked the question is from a life sciences background. So the first thing that you need to start with the Python coding, Python programming knowledge, the basic knowledge. So the Python ecosystem, then you need to start understanding. The number one is the NumPy. NumPy, because the biggest criticism with the Python is that it is slow. It is not scalable. This is where NumPy comes in. So because the NumPy, the background is C language. So that's the reason any arrays that you define in Python, if it takes 100 seconds, NumPy will take 1 second, which essentially means that it is 100 times powerful. So that's why along with Python, you go with the NumPy. Now you have to handle big data. So if you have to handle big data, you need to have pandas. Pandas is the library which enables you to handle all the rows, columns, values, combine the rows, combine the columns, do all sorts of things that you can do it with the pandas. Very powerful, extremely powerful library. So I'm telling you in the order: Python, NumPy, pandas. Then fourth one is the matplotlib. Matplotlib is that doing the visualization, whether it's a scatter plot or box plot or any of the various combinations or of. So it goes in this order: Python, NumPy, pandas, matplotlib. Now this is very basic for handling data science part of it. Now the modeling part of it. So the modeling part, where you are really want to jump into AI, you start with Scikit Learn, which is a conventional traditional machine learning. All the linear regression, logistic regression, support vector machine, all of them are there with the Scikit Learn. So you need to that I showed you the web page, Scikit Learn. And another reason why even if you are not using the conventional machine learning, but you are interested in deep learning, still you need to get some concepts of coding from Scikit Learn. The reason is when you have a database, how you are going to split the training set and test set. We use the utilities. It's called utilities in Scikit Learn. So we make use of the utilities in Scikit Learn for doing deep learning. And deep learning, how we are going to do? Deep learning, we will be doing it with the use of two things. Either TensorFlow enabled with the Keras or PyTorch. So one of them. So what I see as a trend is the academic research community of late, they are moving towards PyTorch. Why I am saying why these two are very powerful, it is like a Coke versus Pepsi. They are the most two competitors. The PyTorch is from Facebook, TensorFlow is from Google. And if you go to YouTube, every year they conduct technology summit, both PyTorch and TensorFlow, and you can start from there. And all the reading materials, all the tutorial materials, all the workshop materials are all available for free. Nothing is expensive. Those days are gone. So like 10-15 years, if anybody tells you that pay me 2.5 lakhs and I'll teach you TensorFlow or PyTorch, don't trust on that. Don't spend money on that. All the best materials are all available for free. That's my advice. I hope this is clear for you. And to combine the AI and the domain knowledge, go into that BioIT World Expo, and that is a very resourceful material. So that's from there. If you start, then the rest of it you yourself will take off. You don't need any guidance from anybody else. I hope things are clear.
Thank you, sir. Correctly pointed by you. BioSolve IT is one of the useful sites where people can look for any of the AI methods or applications in drug discovery. Yeah, and that was actually one of the another question that was there in my list. So I hope that is being answered here. And these were few of the questions which the people had asked, and I would like to thank you once again for your for the elaborative discussions as well as for your comments and viewpoints that you have shared with us. Thank you very much, sir.
Thank you so much.
And may I request now Dr. Mahanta for giving the final concluding remarks for this session.
Hello, sir.
Yeah, thank you, Lizo. Thank you, Dr. Parthiban, for such a very wonderful and informative session. It's very interesting and something which you will have a lot of work to do in near future. So I'll just expect that, sir, if you can find some more time in some other sessions, because I think there's much more things to learn. We can't end here wherever you ended today. So I'll request you to give some more lectures in near maybe coming weeks.
Oh yeah, actually I'm giving a course in IISER Bhopal starting from I think August 17th to 20th or something. I'm taking one full semester.
Oh,
And
okay.
most of this I'm teaching there. I don't know how you can coordinate that stuff. Maybe think about it, because this is not something in 1 or 2 lectures anyone can master. It's actually it's worth doing one full semester, because most of especially the life sciences people, they have to start from Python basics, and it's a long way to go.
Exactly. Very true. So, but still you gave a very good overview of everything. So on behalf of the entire DDH team and the chairman, Dr. G.N. Shastri, Director NIST, I'd like to thank you
so much
for handling this session, and also all the viewers who were there for the session. Thank you so much. So we wind up this session here, and our next session for vertical will start at 6. Thank you, everyone.
Thank you. Best wishes to all the participants. Bye.
Thank you.